Hello, everyone, and welcome to this edition of the GOA-ON webinar series. My name is Michael Aquafreda, and I'm a postdoctoral researcher at the NOAA Ocean Acidification Program. And today I'll be moderating this webinar. This webinar has four sponsoring organizations. First, GOA-ON, the Global Ocean Acidification Observing Network. Second, NOAA, the National Oceanic and Atmospheric Administration. Third, the IAEA OAICC, the International Atomic Energy Agency, Ocean Acidification International Coordination Center, and last but not least, the IOC UNESCO, the Intergovernmental Oceanographic Commission of the United Nations Educational, Scientific, and Cultural Organization. For those of you who are new to GOA-ON, it is a collaborative international network designed to detect and understand the drivers of ocean acidification and the resulting impacts on marine ecosystems. GOA-ON serves as a platform for acquiring and exchanging data and knowledge that is necessary to optimize modeling studies, and GOA-ON also provides key inputs to communities, industries, governments, and global organizations who are seeking to develop action plans, best practices, mitigation, and adaptation strategies to address ocean acidification impacts. GOA-ON has over 860 members from 105 countries, and we welcome any audience member who isn't currently a member of GOA-ON to become a member. During the presentation, all participants are in listen-only mode. You are welcome to type any question into the questions box, which can be found on the bottom of the control panel on the right-hand side of your screen. We'll be moderating incoming questions, and we'll pose them to our speaker at the end of today's session. We also encourage you to post questions and share your insights on the Ocean Acidification Information Exchange. Today's webinar is being recorded and will be available through the GOA-ON YouTube channel. At this time, I'd like to introduce today's speaker. Dr. Elliot Scanes is a Chancellor's postdoctoral research fellow in the Climate Change Cluster at the University of Technology in Sydney, Australia. Elliot completed his PhD at the University of Sydney in 2017 and has since conducted research in Norway, Germany, and more recently back in Australia. His research focuses on the biology of marine invertebrates and their responses to climate change. And without any further ado, I'll now pass it over to Dr. Scanes.
Okay, Michael, thanks very much for the great introduction. I just hope that everyone can see my screen.
Yeah, it looks great.
And great. Okay, well, I'll just proceed from here. And as Michael just mentioned, today I'll be talking about the response of the Sydney rock oyster and its microbiome to rapidly warming and acidifying Australian estuaries. Now, before I go any further today, I would just like to acknowledge the Gadigal people of the Eora Nation upon whose ancestral lands I'm giving this presentation. And I would like to pay respects to the elders, both past and present, acknowledging them as the traditional custodians of knowledge for this land. I would further like to acknowledge the traditional custodians of the variable ancestral lands from which our other attendees join us today, and pay respects to those elders past and present. So today I'm going to start off talking about estuaries and how climate change is manifesting in them, and then go on to how these impacts of climate change may flow onto other organisms, especially oysters. So estuaries are extremely important environments because they are essentially where the ocean meets the land and where freshwater inputs enter the ocean. Here we can see some examples of estuaries in Australia, but they also exist on every other continent except for Antarctica. And we can see that similar estuaries exist around the world, such as on the western coast of the USA and in South Africa. So estuaries are vital ecosystems because they provide a lot of habitat. So almost all seafood that we eat completes part of its life cycle in an estuary, and that habitat that it provides is novel and complex. Estuaries are home to threatened and endangered species. In Australia, some estuaries are also designated Ramsar sites, which means that they are of international importance for migratory birds. Estuaries cycle nutrients, so they store carbon, they allow for the trophic transfer of nutrients from the sediment to the water column, and they feed the food webs of the ocean, almost all the nutrients that are in the ocean have passed through an estuary at some point. And they are the highways from nutrients to be taken from the land into the oceans. Estuaries also provide a lot of social benefits for us humans, such as protecting the shoreline, providing resources for aquaculture and wild fisheries, and also for recreation. And estuaries are diverse and unique. So no two estuaries are alike. So we see just different types of habitat and different types of vegetation occurring in every estuary. So again, every estuary is unique. So today I'll be talking about estuaries in New South Wales. And for those who are not from Australia, New South Wales is a state on the eastern coast of Australia. It is the most populous state in Australia, and it covers about 1,100 kilometres of north to south eastern coastline. The climate of New South Wales is sort of more of a warm, temperate and subtropical in the north, but the East Australian current brings warm water down the east coast of Australia, making the oceans of Australia a more of a subtropical climate. So the government of New South Wales in 2010 started a monitoring program of estuaries within New South Wales called the MER program, monitoring, evaluating and reporting of estuaries. And what this was a large water quality monitoring program of estuaries to collate and synthesize the information that is available. So as part of this program, they classified the estuaries within New South Wales into 5 types. Firstly, there was rivers, which have a wide entrance and are always open to the ocean, and there's 52 of those. Lakes, which are the largest type of estuary, and they're also almost always open to the ocean, and there's 28 of those. Lagoons are smaller than lakes, and the entrance is often closed. And I'll be talking more about that later, but as you can see in this image here, the beach has actually cut off the lagoon from the ocean, and there's 43 of those. Back dune lagoons are similar to lagoons, but they are fed by more groundwater rather than river water. And again, they're often closed to the ocean, and in New South Wales, there's 23. Creeks are the smallest estuary type. They have a short retention time, which means water does not stay within them for long, and they're sometimes closed. So we know that the oceans and the land around Australia is warming. On the left-hand side here, we have the East Australian current, which is the warmer red water being drawn down the east coast of Australia. And we know that this is going to intensify in the future with climate change. And on the right-hand side here, we can see a map of Australia showing that where the land area has warmed since 1970. Anywhere that is red, as you can see on the scale on the right, has actually increased in temperature since 1970. So we can see that large parts of the Australian land mass have also warmed, and a lot of this warming is concentrated in the eastern side of Australia. Now, in addition to the warming and that we're seeing in Australia, the oceans off the east coast of Australia are a warming hotspot, which means that they're warming faster than the global average. So here, if we look at this map of ocean warming around the world, we can see that off the east coast of Australia, there is a spot of greater than average warming, making this an ocean warming hotspot. Now, we've heard a lot about the intergovernmental reports on climate change lately, especially because of the new one that came out yesterday. But in all of these reports, and I checked in the new version, and it seems that this is still the case, there isn't really any mention of estuaries, and this is largely because they're such an understudied system. So we have a pretty good idea of what climate change is doing on the land in Australia, and we have quite a good idea of what it's doing in the ocean. But basically, estuaries are sort of an unknown zone, which is a little bit of a worry considering how important they are for the functioning of the oceans and also for so many of the resources that humans rely on them for. Some previous studies have focused on high resolution data from a single estuary, and none of these are from Australia. One example is there's great time series data for large estuaries such as the Hudson River in New York. However, this is just for one system and doesn't capture how estuaries as a group are responding to climate change. So as part of that statewide estuary health monitoring program that I spoke about earlier, we were able to get temperature, pH and salinity data for the last 12 years in Australia for 166 estuaries, covering 6,200 observations and 1,100 kilometers of north-south distance along the coastline. And you can see here, covering the entire coast of the New South Wales state. All measurements were actually taken by hand. So technicians in the field, scientists were there using handheld instruments taking measurements. And this is largely because estuaries are so small, they're very difficult to measure by satellite, and you get a lot of interference from the land masses around. So really the only way to do this is in situ taking measurements by hand. And this is one of the reasons why estuaries are quite poorly understood, purely because it's so time intensive to take these measurements. In addition to this data, we also collected catchment characteristics for all these estuaries. So all the things about an estuary that we could learn, we learned. So drivers of change such as depth of an estuary, the area that they took up, the flow of water through an estuary, how long it can hold on to water, which is its retention, the vegetation around it, and also the vegetation and use of the entire catchment for every estuary on the land. So as mentioned earlier, this meant we had 52 rivers, 28 lakes, 43 lagoons, 25 creeks, and 23 back dune lagoons. So rather than focusing a lot of effort on one estuary, we focused efforts across multiple estuaries and all types. So we had 166 different estuaries up and down the coast of New South Wales. And instead of putting a lot of effort into one of these, we focused on all of them to try and understand how climate change was manifesting on average across estuaries. So with this time series data, we used linear models to determine significant change in rates in temperature, pH, and salinity for each estuary type. And that type's what I was talking about before with rivers, lagoons, etc. So now I'll just go through some of these results in terms of temperature change over time. And for lagoons, we can see that over the study period, we found on average a 3.65 degree increase in temperature. Rivers increased by 2.79 degrees, back dune lagoons by 1.31 degrees, lakes by 1.07 degrees, and creeks showed no significant change in temperature. So 4 out of our 5 estuary types all significantly warmed, and some of them warmed in excess of 2 degrees over the study period. So this gave us an average of overall for all estuaries, 2.16 degrees increase in temperature over the study period. pH was not measured for the entire study because of it was not originally part of the monitoring program. So our pH data only goes back from 2013 to 2019, but we can see a significant decline in the pH of creeks by half a pH unit. Lagoons decreased in pH by 0.46 units, rivers by 0.32 units, back dune lagoons by the same amount, and lakes by 0.28. So all of our 5 estuary types significantly decreased in their pH over the study period. And this came to an overall average of 0.51 pH units. For salinity, the results were a little bit more varied. So we saw creeks freshened the most by 6.74 salinity units, lagoons freshened by 5.38 salinity units. Rivers actually increased in their salinity by 2.68 units. Back dune lagoons and lakes showed no significant change in their salinity. So again, we can see that the change in salinities depend on the estuary type, and this came to an overall average of 0.97 salinity units, which is a freshening of estuaries on average. However, like I just mentioned, it was highly dependent on the estuary type that we were looking at. So in addition to using these linear models to determine the rates of change, we also use random forest regression models to determine which characteristics of estuaries and their catchments contribute to change. So this is what I was talking about before. We took, instead of grouping estuaries by their types, we actually just took all of the information about every single estuary and fed this into a random forest model to determine which characteristics of an estuary were best predicting the change in the variables. So this is our list of predictive variables that we used to predict change in estuaries in our model. So firstly, we had the days elapsed. So this was the number of days since sampling, the month that the measurements were taken, the retention factor of an estuary, so how much water can it hold on to before flooding, the latitude of the estuary, the size of its catchment in hectares, the average depth of an estuary, which is essentially like its surface area to volume ratio, the total flushing time of an estuary, how long does it take water to travel through it, the percentage of estuary covered by seagrass, and the percent of the catchment which has been cleared for farming or other housing, urbanization, etc. Percentage of the catchment which has been urbanized, and the proportional increase in nitrogen that has occurred from farming and other practices within that catchment. So here we see those same predictors, but now they've been organized in terms of their importance. So in terms of temperature, we can see that the most important thing, sorry, and I'll just go back one step, these have also been color-coded by their type of predictor. So the blue ones are time, green are geomorphology characteristics of the estuary, and the reddy sort of color is the catchment characteristics of the estuary. So we can see here that the most important factor driving temperature in our study period was the month that the measurements were taken, which makes sense because we know that some months are warmer than others. We then have the days since the time was taken, which is also makes sense because we know that estuaries have increased in temperature over time. Thirdly, we have the latitude of site, which is also as expected because we know that the Earth is warmer at the equator compared to the poles. But then we get to some interesting things such as average depth, retention capacity, and total flushing time of estuaries. So we know that other than the top three things, these other characteristics of estuaries are what's driving their temperature change. In terms of pH, we see time in terms of since sampling days elapsed at the top, latitude of site next, month of measurement third, and then we see the total flushing time of an estuary is quite important, and also the percent of the estuary that is covered by seagrass. So we can see that there's, we have the expected ones at the top, such as time and latitude, but then we see some other interesting drivers of pH in estuaries. In terms of salinity, we see two time variables at the top again, days and month of measurement, which is predicted because we know that some months are wetter than others. But then we see the retention capacity of an estuary, so how long can it hold on to water, as a strong driver of salinity, as well as latitude and size of the catchment. So a larger catchment area would catch more fresh water and therefore contribute to the salinity of an estuary. So just to summarize these results, we've shown that overall estuaries have warmed by 2.16 degrees Celsius. And this warming is concentrated in lagoons and rivers. So now just to take a step back and think about climate change in Australia more broadly, here this map, or this graph rather, shows the temperature average for all of Australia every year since 1910. And the blue bars show when a year that was cooler than average, and the red bars show a year that was warmer than average. And so this is our study period here. So we can show, we know that our study has coincided with a warmer part of Australia's temperature history. And similarly, if we look at sea surface temperatures, again, this is the same graph, but for sea surface temperatures in Australian waters, we can see that, again, we've coincided with a warm part of Australian sea surface temperatures. But we found that on average, estuaries had warmed by up to 2 degrees. And if you look at here, this is just the air temperature again. We can see that on average, Australia is warmed by about 1 degree in terms of air temperatures. So on average, the warming in estuaries was double that of what Australia has experienced in terms of air temperature warming. So why might this be? Well, we know that air temperatures and ocean temperatures in Australia warm by about 1 and maybe 1.5 degrees over this study period. But lagoons warmed by 3.65 degrees. So when we think about our drivers, we know that average depth and retention capacity are quite important. So this is what I was talking about before when I was talking about how the mouth of an estuary can become cut off. And we can see in this example here, if you look at Termea Lake, which is a lagoon, the beach has completely cut off the estuary from the ocean. So these sorts of smaller estuaries are shallow, they get cut off from the ocean, and therefore they're able to absorb a lot more warmth than either the ocean or the air. And what this plot shows is the predicted temperature, which is the color coding. And along the bottom axis, we have the flushing time of an estuary, so how long water stays in the estuary for. And along the vertical axis, we have the average depth. So if we just use those two predictors from our data to predict temperature, we get this pattern. And we can see that lagoons sit in this warm area where they have a medium flushing time, so water stays within the estuary for up to around 50 or 60 days. And they're also quite shallow, which means that water is retained, and due to their shallow nature, it absorbs more temperature, more warming per volume area. And we can also see creeks sit in this warming area. However, creeks were just so variable, we found no significant effect. So again, in terms of when we think about acidification and the freshening of estuaries, we found that creeks and lagoons acidified and freshened the most. Now, this map shows the trend in total rainfall since 1970 in Australia, and anywhere that is a yellowy brown color is where rainfall has declined. So we can see in the eastern half of Australia, rainfall on average has declined since 1970. And if we look again, the same map, except now we have below average rainfall as red colors, Eastern Australia in 2018 on the left and 2019 on the right received quite low amounts of rainfall, very much below average, and in some cases lowest on record. And what that means is that when estuaries experience low rainfall, they get cut off from the ocean. And just because I know that this is an ocean acidification audience, the changes that we saw in estuaries in terms of pH are greater than that would be expected by CO2 alone. So while CO2 is likely playing a role in their acidification, there's more likely to be other processes at play here that are causing this acidification in addition to CO2. And one of those has to do with flow rates. So in periods of low flow, estuaries such as lagoons and creeks get cut off from the ocean by a sand barrier or another type of barrier, but they still have small amounts of fresh water flowing in from either groundwater or from the catchment. So while they're cut off from the ocean, they have small amounts of fresh water coming in, which means that they actually are freshening, which is counterintuitive. You would think that in a drought, estuaries would become more salty, but they're actually becoming fresher as they're cut off from the ocean, yet small amounts of fresh water are coming in. And we know that freshwater input into a system like an estuary can also contribute to acidification, whether it be from dissolved minerals or by diluting the bicarbonates that are present already within the estuarine system. So just to wrap up, we know that what the lands are doing and what the ocean's doing, but it seems as though estuaries do not follow the pattern of the land or the ocean, but rather have their own processes driving the impacts of climate change, leading to them doing their own thing. So why can't we rely on oceanic models or land models to predict temperature in estuaries? And this comes down to the fact that these types of models create average effects over a large area, and they ignore small-scale variation, whereas estuaries are essentially that small-scale variation that is being ignored. And of course, there are no estuary-specific models. So by comparing air temperatures and ocean temperatures to our data on estuary variables, a model could be built to forecast estuary change, which is what we're working on. So what does this mean for estuaries around the world? So we know that these types of estuaries in Australia are quite common, and again, we've done extensive analysis on this, but we see similar types of estuaries on the west coast of the USA, in South Africa, and also in the Mediterranean. So it's likely that the processes we're seeing driving climate change in Australian estuaries are also going to be applicable to those similar types of estuaries in other parts around the world. And we know that warming is likely to cause ecosystem shifts, and this is going to be further accelerated by freshening and acidification. Tropicalization is already occurring on the New South Wales coast and around the globe. And we've seen papers such as this, where this image is from, where we see tropical species moving south in Australia and moving north in the northern hemisphere. And these are likely to be entering estuaries and causing tropicalization of these systems. One interesting thing, and a positive out of all this research, was that seagrass was found to buffer acidification. We found that the presence of seagrass was a strong predictor of pH, and those estuaries with a greater proportion of seagrass had an increased level of pH. So this is something to think about going forward. So Australia's estuaries are being altered by climate change as Australia warms and dries, and this change is going to be felt most acutely in small estuaries. This warming will affect the organisms and industries that rely on estuaries. And this is where I want to talk about some of the ecosystems and organisms that we know live in estuaries, and some of my other recent research focused on oyster farming. So here we can see oyster farming occurring in Australian estuaries. And we know that oysters aquaculture is worth $40 million Australian dollars in New South Wales alone. Oysters are a low carbon and highly sustainable food source that is recognised as vital to Australia's future. But oysters can only be grown in estuaries in Australia. And we know that this makes the oyster industry highly vulnerable to climate change, with the incidence of disease currently increasing. So when we think about oysters and disease, there's essentially three main contributing factors. We have the host, which is the oyster, the pathogen, whether it be a virus or a bacteria, and the environment that these two are occurring in. And we know from what I've just talked about for the last half an hour that the environment of oysters is going to be changed, and it's going to change fast. So oysters, like all other organisms, have microscopic animals living within them, be them bacteria, and these form the microbiome. So in oysters, we know that the microbiome is vital for immune function, as well as digestion, amongst other processes. So we wanted to determine how does the warming and acidification predicted and also observed in estuaries for the coming century affect the microbiome of oysters, and what implications this might have for oyster disease. And we also wanted to know, what role does the oyster genotype play in the response of the microbiome to disease? So to test this hypothesis, we set up an experiment where we exposed wild Sydney rock oysters, Saccostrea glomerata, to temperature treatments of 24 and 28 degrees, as well as two CO2 scenarios of present 400 microatmospheres and future 1000 microatmospheres to give a pH of 7.8. We then used Illumina sequencing of the 16S gene to characterize the bacterial communities present in the hemolymph, which is the blood, of our oysters from our treatments. So what we found was that there was 967 different bacterial sequences identified from our wild oyster samples, and 10 bacteria were found in all samples, comprising 31% of all bacterial abundance. So there was a lot of commonality amongst oysters. So just some results here. On the vertical axis, we have bacterial diversity, and on the bottom axis, we have our ambient and elevated CO2 treatments. The blue boxes are our 24-degree treatment, and the red boxes are our 28-degree treatment. And you can see here that at elevated CO2, we saw a significant increase in bacterial diversity, but this effect was somewhat tempered at elevated temperature as well. So already we can see that changing the pH and the temperature of the oyster's environment is affecting the bacterial communities that are present within oysters. This plot is just an ordination plot of our bacterial community compositions. And we can see that the blue circles at the top are our ambient 24 degrees ambient CO2 treatments. And we can see that for the other three treatment combinations, the treatment variables are shifting our bacterial communities. So again, we can see that by warming and acidifying the oyster's environment, we're seeing a shift in the bacterial communities present within the oyster. And we can see that there's just a wide variety of bacteria correlated with different temperature treatments. So on the right, we have 24 degrees and all the different bacteria that are correlated with that. And on the left, we have 28 degrees with far less bacteria correlated. So we can see that different bacterial communities are being driven by these different temperature treatments. So now we know how warming and acidification is affecting the microbiome of wild oysters. We want to know what role does the oyster genotype play in the response of the microbiome. So we've looked at the environment, but now we want to look at those host factors driving the oyster disease and how changing that may affect the disease paradigm. So to determine the effects of genetic variation, we examined how these patterns changed across oyster families bred for aquaculture. So here we have 9 oyster families, and when I say a family, all the individuals within each family share the same mother and father and are not related to the other families. So we can see that genetic variation within families is quite small, but compared to other families, genetic variation among the families is quite large. So again, we use the same experimental design where we have our temperature and CO2 treatments. And again, we used our Illumina sequencing of the 16S gene, except this time, instead of exposing wild oysters, we exposed our 9 families of the Sydney rock oyster to these temperature treatments and CO2. We found that 5,980 different bacterial sequences were identified in our 9 families. So immediately, by increasing the genetic diversity of oysters, we can see that we're increasing the amount of bacteria that we're finding. So increases in genetic diversity are equating to increases in bacterial diversity. Again, some bacteria were found in all samples, showing some commonality amongst the families. But here, this is just an ordination plot of our 9 families of oysters. And we can see that this is just an ambient CO2, 24 degrees normal seawater conditions, that just at these conditions, the families are all quite different to each other. So we can see that the genotype of family is already driving the bacterial communities that are associated with that family. And some of these bacterial members have been found by others. So we know that Mycoplasma and spirochates found in this study have been reported in other parts of the world and other parts of Australia. So these bacteria are quite common in oysters, and they're popping up every time oysters are examined, showing that they are not there purely by chance, but rather these bacteria are important to oysters, or the oysters are important to the bacteria. So this sort of intimidating plot here just shows different types of bacteria organized into their taxa. And along the bottom, we have our temperature and elevated CO2 treatments and our 9 families, which have been labeled from A to I. And when there's a black square, that means that the bacteria are present in 90% of those samples. So just to show quickly, we can see that these Archobacter are present at ambient CO2 in some of our families, but they disappear at elevated CO2. And sort of the reverse, we can see that these Marinophiliaceae are quite common at elevated CO2 amongst some of our families, but again, they disappear at ambient CO2. So we can see that the CO2 patterns are not only driving the shifts in low abundant taxa, but these taxa which are present in almost every sample are being shifted by our CO2 treatments. Here we have the bacterial diversity on the vertical axes and our families along the bottom, and the grey squares are represented by elevated CO2 and the white squares are representing ambient CO2. So we can see that in only these 2 families, D and G, was there a significant increase in bacterial diversity under elevated CO2. And again, this is what we found for wild oysters, although we're only seeing it replicated in 2 out of 9 different oyster genotypes. So again, we can see that the effect of CO2 on bacterial diversity is dependent upon oyster genotype. Here, these ordination plots show the bacterial community composition under either ambient or elevated CO2, and I've just selected the 3 families for which this was significant. So ambient CO2 concentrations are only affecting the bacterial communities of these 3 families. Similarly, if you look at elevated temperature, it is affecting only these 3 families as well. And we found that the shifts in these Malachiobacter and spirochaete are driving a lot of the changes in the affected families. So what does all this mean for the oysters? Well, both those bacterial genera that I just touched on are closely associated with oysters. So they're changing their abundance is likely to have health implications for oysters and oyster disease. They likely perform functions such as part of the immune system. So we can imagine how changing that aspect of the oyster microbiome may have downstream flow-on effects for oyster health and immunity. But we only saw 3 families whose bacterial community was affected by CO2, and 3 families, 2 of which were the same 2 families, affected by warming. So we know that warming and ocean acidification can alter the microbiome, but wild oysters and 3 families were affected. However, the remaining families were not. So this shows that there is the capacity to use this existing genetic diversity and resilience to warming and acidification, potentially for selective breeding and mitigating some of the effects of disease that may be occurring as a result of climate change. So maybe why are some of these families affected but others are not? So we know that microbiomes and host physiology are tightly linked in humans and other animals. The gut is one perfect example. There's lots of experiments where if you can transplant the gut microbiome from one rat to another rat, it can have drastic effects on its physiology and metabolic processes. And we know that the metabolic rate of oysters differs among families. So this is a study that looked at the metabolic rate of different oysters at ambient and elevated CO2 using not Sydney rock oysters, but Pacific oysters. But you can see here that the effects of CO2 on metabolic rate are highly dependent on which family those oysters belong to. So again, we can see the interaction with genotype when it comes to metabolic rate and CO2. So we measured the metabolism of oysters as well, and we can see here in the closed respiratory chamber, this is a scallop, not an oyster, but the same process was used. And we found that species richness of bacteria is correlated with metabolic rate, and this is something that we're going to be investigating further into the future. So thank you for listening to my seminar today, and I'd just like to thank everyone I've worked with and helped out on these projects in the past. And you can find most of the results that I've presented today in these papers on the right-hand side. So again, thank you very much, and please welcome any questions that you may have. Thank you.
Hi, Elliot. Thank you so much for that excellent talk. I'll invite our audience members again to please type your questions into the questions box, and we will get started from there.
Michael, do you want me to stop sharing screen now?
Yeah, that'd be great. I'll take it over. Okay, so just checking the questions box now. And I think we'll give our audience a few more minutes to type in some questions. I actually had a question for you. So I was wondering if there are any forecast models of estuaries in Australia. There's a few of those that are up and running in the United States across estuaries. I'm thinking specifically about like the Chesapeake Bay environmental forecast model that is able to produce nowcasts and also like 2-day forecasts. And it's a real asset for stakeholders like fishermen and aquaculturists. And I'm wondering if there's anything similar to that in Australia.
So when you're talking about forecast, Michael, are you talking about like water temperature and things like that?
Yeah, so that one I mentioned forecasts temperature, dissolved oxygen, and is able to identify hypoxia zones and salinity and pH as well.
Yeah, okay. So I guess firstly, an estuary like Chesapeake Bay is probably 100 times bigger than any estuary that we have on the East Coast or anywhere in Australia. So the amount of variability in our small estuaries is significantly higher than a large body of water where it is a little bit more reliable and predictable. So I'm not really aware of any of these predictors that are available for smaller estuaries. Some of the larger, more oceanic estuaries, I think that these services are available. However, they're mostly based on oceanic temperatures and the influence of the ocean on that estuarine system. Again, like I said, the small nature and the variability that causes can make predicting any sort of even short-term temperature changes within estuaries very difficult. And on top of that, we have over 166 estuaries in New South Wales. So essentially you would need to use a specific model for every single estuary. However, a lot of oyster aquaculture is concentrated in some of our larger, more oceanic estuaries, and perhaps a model could be used in those systems, and that could be quite useful going forward if it's not something that already exists.
Awesome, thank you. I didn't realize they all were so small.
Yeah, well, I mean, Australia is the driest continent other than Antarctica, which means that, you know, low rainfall means that we have small estuaries, and also the geology of Australia, where we have a large mountain range quite close to the coast on the eastern side, means that a lot of the water that falls inland in Australia drains west away from the coast. So the coastal environment only receives a catchment from the immediate coastal strip, therefore causing quite low rain, small estuaries with low flow that can be easily cut off from the ocean.
Very interesting. Thanks for all that. So we have a few questions coming in. The first is, what could the next steps be to determine if the immune system is being impacted? Were all of the oysters disease-free when you started these experiments? And did you look at any interplay between the microbiome and specific oyster pathogens?
Yeah, so in relation to specific oyster pathogens, I assume the questioner is talking about things other than bacterial pathogens, because we were able to see any bacterial pathogens within our microbiome sampling. So things such as Vibrios and other disease-causing pathogens were quite low in abundance. However, we did see in some cases those potentially disease-causing pathogens to increase under some of our climate change conditions. In relation to other pathogens, the Sydney rock oyster is not affected by the oyster herpes virus, which affects specific oysters. So we didn't have any issues with that. And largely, we were aware that these oysters were disease-free because we didn't see any deaths of oysters throughout the experiment. In terms of looking at the immune system, there is some research that shows hemocytes and other immune cells are affected by elevated CO2 conditions. So potentially this is a reason why the microbiome is being altered, because the immune system of the oyster is also being altered. However, more detailed immune studies and perhaps a challenging study where you inoculate the oysters with a pathogen and then expose them to these environments would be useful to determine how disease might manifest under these conditions. One disease that is prominent in Sydney rock oysters is called QX disease, which is caused by a haplosporidian parasite. And the interactions with this and the microbiome under climate change would be interesting to investigate.
Excellent, thanks. So we have another question. This one's actually about viruses. The participant is interested in knowing whether or not you looked at oyster viruses and what consequences there may be for the virome of the oysters, considering climate change.
Yeah, great question. And that is one thing that we didn't look at, viruses, something that is a little bit more difficult to investigate compared to the bacterial communities. And in some ways, viruses don't have the same beneficial functions that a lot of bacteria provide, such as digestion and immunity and things like that. But certainly viruses are an important aspect to the oyster microbiome. Research on the herpes virus that I just mentioned has shown that it is not the virus that actually kills the oyster. The virus causes the oyster's immune system to be affected, and then bacterial infections come in and take over and actually kill the oysters. So a lot of these pathogens act in tandem with each other, and viruses can certainly have flow-on effects for bacterial abundances. So certainly viruses are something that we need to look at. But like I mentioned, for the Sydney rock oyster specifically, we don't have these highly prominent viruses like Pacific oysters do. So a little bit more investigation would be required there.
Absolutely. Following up on that, were you able to identify any specific genes or SNPs or genetic regions that were associated with any of the changes in diversity that you observed, or any of the changes in bacterial diversity?
Yeah, so we didn't look at any transcriptomics or the oyster gene regulation. However, this is partly made a little bit more difficult because there's only a draft annotated genome available for Sydney rock oysters, and it's only become recently available.
Sure.
So unlike other oysters, which are far more, studies are more advanced, such as Pacific oyster, which has a, genome that's been published for almost 10 years now. We're still working on that for the Sydney rock oyster. So to delve into that would take a little bit more work. However, a lot of research on the Pacific oyster, which is a close relative of our Sydney rock oyster, has shown that specific genes can be upregulated and downregulated in correspondence with environmental conditions, and this likely has flow-on effects to the microbiome. So that's certainly something that we need to investigate further, and this will be enabled as we learn more about the genome of Sydney rock oysters.
Great. Hopefully that genome gets published soon. Helps you out.
Yeah, no, I mean, it's a great initiative and something that'll be very useful for, like, Sydney rock oysters form the essential entire aquaculture industry in Australia or Eastern Australia. So that is going to be a really useful piece of knowledge to have.
All right, so I think we're going to take one more question. So before I say the question, I should just mention that a lot of people are saying in the questions box that you gave a great talk. So just want to acknowledge that.
Thank you.
But this question here is, do you think that the oyster microbiome can be used to help oysters become more resistant to acidification and temperature stress?
Yeah, I think that using the microbiome as a tool for oyster selection is certainly going to be quite important. As I mentioned in my presentation, we found that overwhelmingly a lot of the oyster genotypes weren't affected. Their microbiome wasn't affected by climate change. So potentially focusing on those families which appear to be more resilient could have positive effects in terms of oyster disease going forward. And I suppose it has the microbiome and these sort of fields are quite new and not something that has probably been considered in aquacultural selection in the past, well, at least not on purpose. So I think using that as an extra sort of tool in selecting for tolerant oysters is going to be important, especially as we know that incidence of disease is increasing. So focusing on that as a priority might be quite important and help oysters in the future.
Awesome. Well, thank you again, Elliot. At this time, I'd like to just say to all of our audience members, thank you for joining us for this edition of the Go-On webinar series. If you have other questions for Elliot, you could post them on the Ocean Acidification Information Exchange. And finally, before we leave, I'd like to remind everyone that Go-On will be hosting Ocean Acidification Week 2021 from September 13th through the 17th. We have dozens of talks lined up. More information about this can be learned at the goon.org website, and we hope to see you all there. Once again, Elliot, thank you so much for joining us today. Thank you to our audience, and we'll see you next time.
Great, and thanks to everyone for tuning in today.